All right. Hello, everybody. Fawzayat asks if the exam will be online, and the answer, Fawzayat, is yes. For now, the exams will be online. We'll have one actually Tuesday. I'll put out a study guide a little bit later today, and yeah, thank you for the question. All right, so I'm gonna jump in here. Actually, can you guys hear me? Let me double check there. Check. Okay, good. Looks like you guys can hear me. Um, all right. So, jumping back in. Wait, I gotta pull up the screen. Hold on. There we go. We left off having finished the granulocytes, the basophils, the eosinophils, and the neutrophils. Today we're going to talk about the agranulocytes, including the monocytes and the lymphocytes. We'll move on to platelets, talk about plasma, then we'll talk about blood clotting. Yeah, and I think blood groups will finish off the day. All right, so monocytes, as you can see, are large. They are the largest white blood cells. All right, they're sort of spherical in shape, and their nucleus has sort of a kidney shape here. Sometimes it looks a little like a horseshoe. Um, so they leave the bloodstream and become macrophages. And macrophages, you probably remember, are the white blood cells that they reach out, they engulf a bacterium, they surround a bacterium, they bring it into the cell, and then they destroy it with lysosomes. Um, yeah, so they bring it into the cell, and they're going to uh, use acid to destroy the bacteria. They'll also use digestive enzymes to break them down. And they can live for weeks or months. They also phagocytize. Phagocytize, by the way, phagocytization is the process of engulfing bacteria or dead cells or debris, bringing them into the cell. And they will engulf not just bacteria, not just fungi and other invading pathogens. They'll also attack dead cells and debris just to clean up your tissues, like the microglia in the brain. All right. Let's see. Study guide helps. Well, thank you. I'm glad they help you. We'll have that out later today. So the monocytes you can see here highlighted in blue. You can see its horseshoe-shaped nucleus here. Sometimes it looks like a kidney. Here it looks like a horseshoe shape. Um, yeah, over here, of course, again, they show you the erythrocytes in blue, and then you see one platelet here. So next up, we have the lymphocytes. The lymphocytes are the last of the blood cells to talk about. And they're slightly larger than red blood cells, so that makes them the smallest of the white blood cells. You see they're roughly comparable in size to the red blood cell. Red blood cells, as far as human cells go, again, are pretty small. So they have a large spherical nucleus, which takes up most of the cell. They are again agranulocytes, and meaning they do not have granules. And then there are T cells and B cells. They're the major types. T cells grow up in the thymus, that is, they mature in the thymus, and then B cells mature in the bone marrow. Both of them are actually produced in the bone marrow, but B cells will remain in the bone marrow and mature there. T cells will move to the thymus to mature. Um, and so we could pretend that's where T cell and B cell actually get their names. That's the easy way to remember. T for thymus, B for bone. B actually stands for bursa of the rooster's comb, which is where they were first discovered. But that's just a little trivia factoid for you. You don't need to remember that. They mature in the bone. The T cells directly attack pathogens, also tumor cells. The B cells produce antibodies and release them. 25 to 33% of all leukocytes are going to be lymphocytes, and these lymphocytes can live for years, and that's important. Um, we have what are called memory T cells and memory B cells, and those are the ones that are going to stick around and continue to, we could say, educate the immune system about past threats. So when you get immunized as a child against diseases, you're going to have memory B cells, for instance, that have seen um, the pathogen before. Let's say chickenpox. Let's say you get the chickenpox vaccine. You're going to have B cells that were exposed to parts of the chickenpox virus, the varicella virus, and they're going to remember it and then stick around for years. And then later, when you're hanging out with a friend who has chickenpox, some of those little viruses are going to make their way over into you, and the infection is not going to take root because you have memory B cells sticking around who quickly recognize the threat and alert the immune system. All right, so diapedesis is the process of white blood cells leaving the bloodstream and entering the tissues. This is done by the monocytes, which become macrophages. Also, neutrophils will frequently do this, leave the bloodstream to enter the tissues. Remember, neutrophils are the first ones on the scene. Um, Good. Phagocytosis, we mentioned that's the process of engulfing or surrounding a bacterium or pathogen or even cellular debris, bringing it into the cell and then dissolving it, breaking it down. The inflammatory response is a restriction that spreads, I'm sorry, it's a reaction that restricts the spread of infection. It's promoted by basophils and actually most immune cells. Most immune cells are going to be able to release this. Neutrophils and monocytes, yeah, especially. Um, anyway, don't worry about that. The basophils, though, they secrete heparin, which is a blood thinner, and then histamine, which is going to involve swelling and make the blood vessels more permeable, or in other words, more leaky, allowing cells to slip out. If you look at the diagram here, you can see a cross section of a capillary, and you can see the wall of it is made up by this endothelial cell. This endothelial cell is really a simple squamous epithelial cell. And there's a gap between the two cells here. And the white blood cell, monocyte or neutrophil, is able to slip through this small gap here. All right, now that process is aided by heparin and histamine. Histamine especially makes the vessel more leaky. And positive chemotaxis. Taxis is movement, chemo is chemical, so this is chemical movement in the direction of a cytokine or other chemical being released by damaged cells. So here we have a splinter in the skin. It is pierced through the stratum corneum. All right, the other 3 to 4 layers of the epidermis as well, down into the dermis. Okay, and then down here in the dermis, uh, you can see there's blood being released because, of course, the epidermis is avascular. So if you see blood, obviously you've gotten into the dermis where we have lots of blood vessels. And so, anyway, the splinter, when it comes in, it brings with it bacteria. It has punctured your barrier. Remember, the skin's a barrier. It's punctured that barrier and allowed bacteria to just bypass straight into the dermis. All right, the bacteria here are going to multiply, And then neutrophils are going to find their way into the dermis. Okay, they're the first responders, first ones on the scene, especially in bacterial infections. And they're going to engulf the bacteria, phagocytize them, and then break them down. And as we continue to fight this infection, the neutrophils will die, and dead neutrophils piling up, that's actually what we call pus. Pus is mainly dead neutrophils. All right, so the neutrophils clear out the infection eventually, and the skin is able to close up and heal. All right, so your white blood, excuse me, your white blood cell count. This is a procedure where we take a sample of blood and we look for the number of white blood cells. You can do this on a microscope slide. It's kind of old-fashioned. You can look for it other ways in modern labs, but the count per microliter should be about 3,500 to 10,500 per microliter. A high white blood cell count is anything over 10,500, anything over this range. And you see this with acute infections, vigorous exercise, or great loss of body fluids. Now, we say great loss of body fluids. Uh, Basically, if you had 10,500 white blood cells per microliter of blood, and then you lost a lot of your blood volume or the fluid in the blood. Let's say you were dehydrated, you'd been working in the sun all day, not drinking water. What that means is that water leaves the blood through sweat and it becomes thicker and more concentrated. So dehydration can lead to a greater concentration of white blood cells in the blood, even though the total number hasn't increased. That can actually throw off a white blood cell count. Leukopenia is a low white blood cell count, anything below this range, anything below 3,500 per microliter. And that can be caused by typhoid fever, flu, measles, mumps, chickenpox, AIDS, polio, or anemia. A differential white blood cell count lists the percentages of each type of leukocyte. And this is important because we know, for instance, if neutrophils are increased, you're probably looking at a bacterial infection, right? Helper T cells, they decrease during HIV infection. So pus isn't infection leaving the body necessarily. What it is it's a sign that the body is responding to an infection. So the neutrophils, they come in and they attack the bacteria, and they are going to, as a process, die. And so the neutrophils keep coming in, they keep attacking the bacteria, they die, and the dead neutrophils just keep piling up, and that becomes pus. Why are you more dehydrated in places of higher elevation? Hmm. Well, I'm not sure about that. If I had to guess, it would have. Hmm. I think maybe the air would be drier. I'm just gonna throw something out there because I don't know. I would guess the air would be drier. See, we lose water actually every time we exhale, and dry air will dry out our mucus passageways in the nasal cavity. Um, they'll also draw water out of the lungs. So, I don't know, that would be my guess. Um, to Kadra, will the exam be online or will it be in class? It will be online. At least the first one will be. And okay, so we'll get back to this. Leukemia is a cancer of the white blood cells, and it can be classified as acute, which is where the symptoms appear suddenly and progress rapidly, or it can be chronic, where it begins more slowly and can remain undetected for months or years. Lymphoid leukemia is a cancer of the lymphocytes, and it's produced in the lymph nodes. Myeloid leukemia is a cancer of the granulocytes, which are produced in red bone marrow. Symptoms of leukemia include an excess number of white blood cells, as well as fatigue, headache, nosebleeds, fever, respiratory infections, bone pain, bruising, and slow blood clotting. I'm looking at a number of those symptoms, and it sounds like a lot of heparin's being released. Yeah. Treatments for leukemia include traditional cancer treatments such as chemotherapy and drugs that target enzymes specific for cancer cells, bone marrow, or stem cell transplants. But stem cell transplants as a treatment are pretty extreme. In order to do it, you actually have to irradiate the patient and completely destroy their bone marrow so that you can transplant bone marrow from somebody else. All right, so moving on, our last blood cell type thing is the platelet, also known as the thrombocyte. And platelets are cytoplasmic fragments of megakaryocytes. We saw a picture in the last lecture on that diagram of all the different cell types. Megakaryocytes are really big cells. Megakaryo means giant nucleus, right? And anyway, the megakaryocytes, they grow up big, they explode, and then all the little cellular fragments that are left over, those become the platelets. They're produced by the hemocytoblasts in response to the hormone thrombopoietin. Thrombopoietin stimulates the production of thrombocytes. The hemocytoblast becomes the megakaryocyte, and the megakaryocyte becomes the thrombocyte, or the platelets, cellular fragments. They lack a nucleus because they're just little cellular fragments, and they're less than half the size of a red blood cell. Now, when you do a blood smear or other blood test, platelet counts should be in the area of 150,000 to 350,000 per microliter of blood. Now, they help in maintaining hemostasis by stopping bleeding in damaged blood vessels and sticking to broken surfaces. They also release serotonin, which causes smooth muscle in the walls of the broken blood vessels to contract. All right, so here we see a megakaryocyte highlighted in blue here and over here. And we also see the nucleus of the megakaryocyte. Here we see platelets highlighted in blue on the right. Okay, you can see all these tiny little fragments, big clump of them right here. Okay, big clump of them, but all those tiny little fragments. So here's a summary of everything we've discussed so far in regards to red and white blood cells. I'll leave that there for a second. You can freeze it if you want to read it, but all right, so blood plasma. All right, so far we've been talking about the formed elements, all of the blood cells and the platelets. The remaining portion of the blood is plasma, and it's the largest portion of the blood. It's at about 55% of its volume. Remember, the red blood cell volume was about 45%. For males, the hematocrit was about 45%. And blood plasma is going to be 92% water. 92% of the plasma is water. And it contains organic and inorganic chemicals. It is also responsible for transporting nutrients, gases, hormones, and vitamins. So the most abundant proteins produced, the most abundant proteins in the plasma are the albumins we mentioned, the globulins, and then fibrinogen. All right, now 60% of all the protein in plasma is albumin, 36% of the protein in plasma is globulin, and then 4% is fibrinogen. Much smaller number, but still an important protein group. The albumins are important. They help maintain the osmotic colloid pressure. And what that means is when we talk about blood vessels, I think next Thursday we'll probably start talking about. Don't hold me to that. I think maybe it's next Thursday. Talk about vasculature and how fluids and other substances are exchanged in the capillary beds. Albumins are going to help draw fluid out of the tissue and back into the bloodstream. Albumins are also important because a lot of drugs, hormones, and other substances that are not water soluble and can't be transported in the blood on their own, they can bind to albumin and be transported through the blood. Uh, Good. Globulin. Globulins include the alpha, the beta, the gamma globulins. There's also the mu, epsilon, and delta globulins, but the main ones are alpha, beta, gamma. And the alpha, beta, gamma globulins, they help transport lipids and fat-soluble vitamins, the alpha and the beta. The gamma globulins make up antibodies in the immune system. So, gases and nutrients transported by plasma. The most important ones are oxygen and carbon dioxide. Now, oxygen, you're not going to find a lot dissolved in the plasma. There's some oxygen dissolved in the plasma, but it's not a lot. The majority of it is held by the hemoglobin and red blood cells. You're also going to find carbon dioxide in the blood plasma, and technically you won't find a whole lot of carbon dioxide in the blood plasma either. What happens is, so if you want to split hairs, sure, there's a bunch, but it's not in the form of carbon dioxide. When carbon dioxide meets with water, it's going to join with water and become a molecule called carbonic acid. And then carbonic acid loses a hydrogen and becomes bicarbonate. And so carbon dioxide will mostly be found in the form of carbonic acid or bicarbonate, really bicarbonate, in the blood. Now this process is reversible. You can actually convert bicarbonate back to carbonic acid, and then you can convert carbonic acid back to carbon dioxide and water. And this process actually is going back and forth constantly in the plasma. Nutrients that are transported in the plasma include obviously amino acids, simple sugars, nucleotides, and lipids such as the fats or triglycerides, phospholipids, and cholesterol. All of the nutrients really are going to be transported in the blood. Non-protein nitrogenous substances are molecules that contain nitrogen, but they're not proteins. Most of these need to be excreted in the kidneys, and high levels in the blood indicate kidney failure. So the kidneys are a filter that cleans the blood, basically, turns blood into urine, sort of. They're filters that we could say clean the blood. Anyway, if the kidneys aren't working, then the waste products and toxins are going to accumulate in the blood. And so high levels of NPNs indicate kidney failure because they're not being excreted the way they should be. In the plasma, NPNs include urea, which is a product of protein breakdown. When we break down proteins for energy, we produce urea as a byproduct. And it makes up about 50% of the non-protein nitrogenous substances. There's also uric acid, which is a product of nucleic acid catabolism, breaking down nucleic acids. And when uric acid accumulates, it can actually cause a condition called gout. Uric acid accumulating in the joints, especially of the big toe, commonly the big toe, causes the condition gout, which can be painful. Amino acids. Amino acids are technically not proteins. They're building blocks for proteins. They're also the product of protein digestion. When we break down proteins, that will release amino acids. Creatine, which stores energy in a phosphate bond. You may have, if you've ever been to a health food supplement store like GNC, you've probably seen creatine on the shelf. You might have taken it yourself. And creatine, what it does is our natural endogenous creatine, which is found in our muscles, it's like a holder for phosphate bonds. In other words, it'll hold on to a phosphate, and then when ATP gets used up, it can slap that they can slap that phosphate back onto ATP to charge it back up. Um, yeah, so it increases our energy storage capacity, our immediate energy storage capacity. Um, yeah, that's enough about creatine. So creatinine is a product of creatine metabolism. Creatinine is a commonly used marker for protein function. It should be excreted, and if we're finding elevated levels in the blood, then we know there's an issue with the kidneys. All right, BUN, blood urea nitrogen, also indicates the health of the kidney, and if the levels are too high, obviously you're not excreting urea in the normal quantity. All right, due to some impaired renal function. Plasma electrolytes. Remember, electrolytes are compounds that ionize and dissolve in water. So that includes like sodium chloride, which splits into positively charged sodium and negatively charged chloride ions. Um, some common ones we find are, of course, sodium and potassium. No surprise there. We talked about them extensively in A&P1. You've also got the calcium, which is not just used for making bones. Remember, we also use calcium in skeletal muscle contraction. Magnesium, which is needed for making certain proteins. Chloride, which typically comes with the sodium. We got the bicarbonate. Bicarbonate ions are important. They're a buffer in the blood. Basically, when we release hydrogen ions into the blood through as a result of digesting our food, that's going to acidify the blood, and bicarbonate can actually accept a hydrogen ion and de-acidify the blood, sort of. It's we would say buffering. It's a buffer that prevents changes in the pH of the blood. We got phosphate, sulfate. All right. So sodium and chloride are the most abundant electrolytes. We eat a lot of table salt, sodium chloride. Hemostasis is the stoppage of bleeding, and so actions that can limit or prevent blood loss include blood vessel spasm, also known as vascular spasm, platelet plug formation, and then blood coagulation. So these mechanisms are most effective in small blood vessel injuries, especially arterioles and capillaries. Vascular spasm can't be done in a capillary. It can be done in an arteriole, though. So it's stimulated by cutting or breaking the small blood vessel, and the smooth muscle, which surrounds the blood vessel, which surrounds the arteriole, will respond by constricting and contracting rapidly. And this slows the blood loss. The ends of the vessel may even close completely, which will stop blood loss. And it's triggered by the stimulation of the blood vessel wall and pain receptor reflexes. The response can last a few minutes, but the effect continues for 30 minutes because smooth muscle, while it's slow to contract, it'll typically stay contracted longer than other muscle types. And so by that time, by 30 minutes, a platelet plug will form, blood will coagulate, and the serotonin from the platelets will cause vasoconstriction. Vasoconstriction is constriction of the blood vessels. So platelet plug formation, here we see, wish there were a way to temporarily turn off my email. Anyway, platelet plug formation. We can see here that the blood vessel wall has been ruptured, and we have platelets traveling through the blood, and they actually stick to sticky surfaces, to rough surfaces. The lining of the blood vessel is smooth. All right, and so the platelets will not stick there. But if there's been a rupture, they actually can stick to the rough ends. All right, and then they form what's called a platelet plug as they stack up and accumulate. When they stack up and accumulate, um, they can block the flow of blood and then prevent blood loss. Brittany, does blood type affect the plasma? Well, one immediate effect, or one effect that you will definitely see, is the presence of certain antibodies. And we'll talk about that a little bit later. But if you have, let's say, blood type A, you're not going to produce antibodies that attack blood type A. And again, we'll talk about that uh, at the end. I believe, yes, that's later in the lecture. Other than that, I can't imagine it making much difference. Although somebody may have found something. I'm not familiar with any. I'm sorry. Ashley Barter. I had seen a patient that had lost muscle use in his leg because of shooting up in the back of his knee. Is this? He lost muscle in his leg due to shooting up in his knee related to blood clots in a vessel or artery. Wow. I don't know. What was he shooting up? Like heroin? Huh. Back of his knee, blood clots in a vessel or artery. I don't know. I think not. I just, without knowing more, I would almost bet it was due to infection or something like that. Yeah. I'm sorry. I appreciate all these off the wall questions, Ashley. I have no idea though. Wow. Blood clots. So we can talk about it though. Blood clots, large blood clots would block blood flow to the muscle. Muscle can go without oxygen for short periods of time. Mm, it's not as sensitive as, for instance, the brain. The brain only has like 30 seconds really before it's in trouble. Muscle, couldn't go forever without blood though. Yeah. So let's talk about the character House. The character House has, and we'll probably talk about this again when we get to the vasculature, but he had what's called a deep vein thrombosis, which is a blockage in one of the deep veins that feeds the quadriceps muscle. The fear there, let's see. Oh, that was it. They removed his quadriceps muscle because the fear was that the clot would move to his heart or lungs or brain. That would have been bad. Um, all right. I'm sorry. I just can't come up with an answer. I was just thinking out loud there. Anyway, back to blood coagulation. It's the most effective mechanism of hemostasis, where we form a blood clot in a series of reactions, in which each activates the next. This is called a cascade, oh, excuse me, where we see one reaction activate the next, 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 activate the next. That's a cascade. So it's initiated by two different methods. They can be extrinsic or intrinsic clotting mechanisms. Many chemicals that are used in coagulation are called clotting factors, and vitamin K is necessary for the function of some of these clotting factors. Coagulation depends on a balance between procoagulants and anticoagulants because you have actually both in the blood. And then well, uh, the major event is conversion of soluble fibrinogen to insoluble threads of fibrin. Insoluble threads are going to clump up and then trap the blood cells like a net. So here we've talked about these methods, the vascular spasm, platelet plug formation, and then finally blood coagulation. All right, now with blood coagulation, again, we have the extrinsic or the intrinsic mechanisms. The extrinsic mechanism would be something like damage to a vessel or tissue. Okay, and then intrinsic mechanism would be something like blood contacting a foreign surface, like the outside of a blood vessel. They're both initiated by different factors. We've got tissue thromboplastin with the extrinsic mechanism and Hegman's factor with the intrinsic clotting mechanism. But after this point, there are a series of several reactions that are initiated by prothrombin activator in both. And then we see the conversion of prothrombin to thrombin in both, and then thrombin eventually leads to the activation of fibrinogen, converting fibrinogen to fibrin, which formally forms the clot. So here we see the extrinsic mechanism. There's tissue damage. The intrinsic mechanism, which is blood contacting a foreign surface, like the sticky, rough outside of the blood vessel. And that's going to cause the release of tissue thromboplastin or the activation of Hegman factor. And we can ultimately skip a couple steps and then come back down to factor 10, which is activated and in turn activates prothrombin activator. Okay. Prothrombin activator converts prothrombin to thrombin. Thrombin converts fibrinogen to fibrin, and then fibrin is finally able to form a clot. We don't have to remember all of the factors. An ER nurse or future physician probably should, but for us, we just need to remember that ultimately the two pathways are going to converge on factor X, which activates prothrombin activator, converts prothrombin to thrombin. Thrombin converts fibrinogen to fibrin, and then fibrin finally forms the fibrin clot, which catches the blood vessels and stops blood flow. There are a lot of clotting factors here, and again, we don't need to remember all of them. This chart's good. This one back here, slide 49. All right, so the fate of blood clots. After a blood clot forms, it's going to retract and pull the edges of the broken blood vessel together, which squeezes serum out of the clot. Serum, by the way, we didn't mention earlier, this is plasma minus all of the, minus fibrinogen and the clotting factors, but most proteins. It's mostly water. Platelet-derived growth factor is going to stimulate the smooth muscle cells around the arteriole and the fibroblasts to repair the damaged blood vessels. Plasmin eventually will digest the fibrin and dissolve the blood clot. Remember, in blood coagulation, we were trying to form this mesh network, this net of fibrin, okay, which captures the blood cells and then forms the clot. And then we are going to dissolve the fibrin threads with plasmin. So a thrombus is an abnormal blood clot that forms in a blood vessel. And an embolus, like a pulmonary embolism, right, is a blood clot that travels through the blood vessels. The embolus is a thrombus that is moving. In other words, a thrombus is an abnormal blood clot, and embolus is a traveling blood clot. And if that blood clot becomes lodged in vessels that are going to feed the lungs, the heart, or the brain, that can pretty quickly result in death. So a pulmonary embolism blocks blood flow to the lungs, A blood clot reaching the coronary arteries is going to cause myocardial infarction, which is a blockage of blood flow to the heart. Then the heart stops, you go into cardiac arrest, and that's called a heart attack. A blood clot traveling through the blood to the brain, if it blocks one of the vessels in the brain, that will cause a stroke. So thrombosis is a blood clot in a vessel supplying a vital organ, such as the brain or heart. We mentioned earlier Dr. House from House MD had a deep vein thrombosis. Infarction is the death of tissues that have been blocked. So in a myocardial infarction, you block blood flow to the heart muscle itself. Remember, blood that flows through the heart doesn't actually feed the heart. The blood that the heart pumps doesn't actually feed the heart. The heart has its own blood vessels which feed the heart, and if they become blocked, then you get the death of tissues after the blockage point. That's infarction, hence the name myocardial infarction. All right, embolism is where we see a blood clot that travels and then gets blocked in a blood vessel, such as a pulmonary embolism in the lungs. If we block the blood vessels leading to the lungs, then we're not going to be able to exchange carbon dioxide for oxygen, and it's even though we're breathing, it's kind of like we're suffocating. And then atherosclerosis is the accumulation of fat in the arterial lining. And that can sometimes cause abnormal clot formation because you remember the intrinsic factor, the intrinsic clotting mechanism, is caused by platelets and blood vessels, blood cells coming into contact with rough surfaces, right? Sorry, it's caused by platelets coming into contact with rough surfaces. Um, Normally, the lining of an artery is smooth. The lining of blood vessels is smooth. But if you get these plaques, pictured in yellow on the right, you get these plaques, you get not just a narrowing of the blood vessel, you also get all these rough surfaces produced, and those rough surfaces promote blood clot formation. Deep vein thrombosis, again, we mentioned, where a blood clot forms due to pooling of stagnant blood. It's mainly in the femoral or popliteal veins or the deep veins of the pelvis. A serious complication of deep vein thrombosis is a pulmonary embolism, where the clot forms in the vessels in the leg but then travels to the blood vessels in the lungs and blocks blood flow. And this occurs with periods of prolonged immobility, like on an airline flight where you can't move for 13 hours. Let's say you're flying to Tokyo or something. Can't move for 13 hours. Not really. Something you can do to prevent deep vein thrombosis on an airline flight is actually, yes, get up and walk around. Getting up and walking around contracts skeletal muscle, which actually pushes on the veins in your legs and will promote the movement of blood out of the legs and back into the arterial side of the blood flow. We'll talk about all that in the cardiovascular unit. All right, sorry about that. So the smooth lining of blood vessels discourages the accumulation of platelets and clotting factors. As a clot forms, fibrin adsorbs or latches onto thrombin and prevents the clotting reaction from spreading. Antithrombin will inactivate additional thrombin by binding to it and blocking it from acting on fibrinogen. So some cells, like basophils and mast cells, secrete heparin, which is an anticoagulant and blocks the clotting reaction. All right, so probably the hardest part of this lecture is in my opinion, is the clotting factors. We're moving on to ABO blood groups, which consistently confuses people, but it's probably the easiest thing in the lecture. So we'll see if we can tackle this in as simple a way as possible. So, in 1910, the ABO blood groups were identified. Their gene was identified and explained the observed blood type incompatibilities. Before 1910, they were doing transfusions, and then just every once in a while, somebody died. And I was like, well, this saves more people than it kills, so we're gonna keep doing it. And and they figured out at some point that some people couldn't get certain blood from certain other people, and they couldn't figure out why. They identified the ABO blood groups in 1910. Today, there are over 31 different genes that are known to contribute to the surface features of red blood cells. These are the antigens on the surface of red blood cells and determine compatibility between blood types. So an antigen is any molecule that can evoke an immune response. I want to say can evoke an immune response. Here it says does, essentially, but can is better. If the immune system finds a foreign antigen in the body, one that doesn't belong there, it's going to produce antibodies against that antigen. Antibodies are proteins that react against a specific antigen. Okay, these are proteins produced by the B lymphocytes. Some B lymphocytes will keep antibodies on their cell surface, others will release them into the bloodstream. Plasma cells, especially, will release them into the bloodstream. In an incompatible blood transfusion, donor red blood cells evoke an immune response in the recipient, and the antibodies in the recipient's plasma agglutinate or clump up around the donor red blood cells. All right, so agglutination is a clumping up of red blood cells, and it occurs when antibodies are latching onto the antigens on the surface of red blood cells. Okay, and so let's say you had a sample in your hands of blood type A, you know, and you wanted to know if it was blood type A. What do? Well, you go get the antibody for the A antigen. The antibody for the A antigen will bind to and clump up around the A antigen. All right, and so you add the A antibody to your blood sample, and if you see this agglutination, if you see this clumping up, what you're seeing is um, the A antibody binding to the A antigen. All right, if you don't see clumping up, it means the A antigen is not there, and so this blood sample is not blood type A. If you do see the clumping up, it means the A antigen is there, and so we are looking at blood type A. So there are over 33 known antigens on the surface of red blood cells, but only a few can really evoke a serious transfusion reaction. We talk about the ABO groups and the RH groups because they can evoke serious transfusion reactions. All right, so blood typing and matching from serology to DNA chips. Accurate blood typing and donor recipient matching is essential to prevent those blood rejection reactions in a transfusion. The human blood can be classified into 31 major groups based on antigens on the red blood cell membranes. There are also many subtypes for each type. All right, serology has been used for many years to determine blood types based on identifying antigens. All right. Basically, we used to do what's called serology. Let's see if we have a picture of that. We don't. I'll pull one up. We used to do what's called serology. Now they do it on a DNA chip to determine your blood type. That's the short of that. Let's go. Let's pull up a picture. picture? Yeah, so you could take your blood and put a drop of it on the microscope slide, okay? And then you could add to it the antibody for A, the antibody for B, or the antibody for the rhesus factor. And if you see the clumping up here, that means it's positive. And so you see this clumping up in this sample is B, blood type B. Looks like they're just doing A and B here. So they do 2 drops per person. They're doing 2 drops per person. And then on the left, they add the A antigen. And on the right, they add the B antigen. Okay. And we can see this person does not have A antigen or B antigen in their blood, so they have to be blood type O. This person has no A antigen, but they do have B antigen, so they're blood type B. This person has blood type A because they have A antigen. You can see this clumping up. That clumping up only happens if the antigen is present. So they have A antigen, no B, so they have blood type A. Then down here you see clumping up in both samples, so they're blood type AB because they have both A antigen and B antigen. Here's a cartoon version of that diagram. All right. All right. If you use the, let's find a better picture. Here we go. We don't have any for the rhesus factor here. RH. Anyway, let's go back to the lecture. We've seen enough of that. So if you have, again, blood type A, that means your red blood cells have the A antigen on their surface. If you have blood type B, you have the B antigen on the surface of your blood cells. And then if you have blood type O, then you have neither A or B. Okay. In other countries, they actually call blood type O blood type 0, which is probably a better name because there are 0 antigens, right? No A or B. Here's the prevalence of certain blood types in different ethnic groups in the United States. Just something interesting to look over. We don't really need to memorize it. So here you see a blood cell with the A antigen. Someone who has the A antigen is going to produce antibodies against A? No, they're going to produce antibodies against B. They will produce anti-B antibodies. Now over here, someone with blood type B, they have the B antigen. What kind of antibody are they going to make? Is it going to be anti-B? No, it's going to be anti-A, right? You produce antibodies to attack foreign blood cells, all right? And so you're not going to produce anti-B over here because those antibodies would attack your blood cells. You want anti-A to attack foreign blood cells if they somehow make their way into your bloodstream. And down here, this person has type AB blood. That means they have the A antigen and the B antigen. They will produce no antibodies against those antigens, right? Because any anti-A or anti-B antigen, I'm sorry, any anti-A or anti-B antibody would attack their blood cell, and that's not good. Now over here, a person with blood type O has no antigens on their cell surface, at least none of the A or B antigens, okay? And they're going to produce antibodies against both A and B, all right? So you're basically, here's the short of it, you're always going to produce antibodies against whatever blood type you don't have. If you're B, then you're anti-A. If you're A, then you're anti-B. If you're AB, well, you've got them both, so you can't be against any of them. And if you're O, you don't have any antigens, so you've got to attack both A and B antibodies. I'm sorry, make A and B antibodies that attack A and B antigens. Agglutination is this clumping reaction where we see the antibodies binding to the antigens, which causes the red blood cells to clump up. We can see a kind of zoomed in photo of what that looks like under a microscope here. All right, so who can donate to who? Type O is the universal donor. Why? Because they don't have, go back one, they don't have any antibodies. And so if you think about the recipient, think about transfusions from the recipient's perspective. If you are receiving blood from somebody, all right, if that blood has no antigens on it, then it doesn't matter what blood type you are, your immune system won't recognize it as foreign, and so you're fine. You won't have any antibodies to attack it, right? Because there are no antigens here. So people with blood type O can give to anybody, or in other words, anybody can receive from blood type O because blood type O does not have antigens on the surface. Now, interesting, the other side of the spectrum, we always talk about the universal donor. I remember being a kid and we would talk about people's blood types and they'd talk about blood type O like it was the best, right? You're the universal donor. Well, it's good to help people and it's good to be a donor, but just from your own perspective, if we can be selfish for a minute, people with blood type AB are the universal recipients, which is pretty cool too, right? They can get blood from anybody because they're not producing antibodies against anything. So it doesn't matter what blood cell or what blood type you've got, A, B, AB, or O. People with blood type AB can receive from anybody. Now, there's one thing we're leaving out here. Now, you've heard, I'm sure, whenever you talk about blood, you talk about it being B positive, B negative, right? Where's that positive and negative come from? That comes from the rhesus factor. And if you have it, we say you're positive. If you don't have it, we say you're negative. It's that simple. If you have it, you're positive. If you don't have it, you're negative. And this is just a different antigen on the surface of the cell. It's a different gene, different antigen, but it can also cause serious reactions. All right, so good. If you are Rh positive, you will not produce antibodies against the Rh factor, or antigen D as it's also called. You will not produce antibodies against that. If you're negative, you will produce antibodies against it, right? If you don't have it, you'll produce antibodies against it. If you do have it, then you won't produce antibodies against it. All right, and so the rhesus factor can actually cause a rather serious condition called erythroblastosis fetalis, also known as hemolytic disease of the newborn. And so we see that here. The mother is Rh negative. She's rhesus negative. So we could say she's A negative, B negative, AB negative. It doesn't matter. She's negative for the rhesus factor. But the fetus is positive, right? Rh positive. So the father of the fetus had apparently positive blood. Oh, the positive and negative, by the way, when it comes to the rhesus factor, that's a. You guys probably haven't had genetics. It's a dominant gene, so you only need one copy to be positive. Anyway, so the father was positive, and moving on. During pregnancy, it's fine. The blood doesn't cross the placenta, and so the mother's immune system doesn't recognize the positive antibodies on the blood of the fetus. Now, during childbirth, some of the mother's blood and some of the fetal blood may mix. Some of the fetal tissue may enter the mother's bloodstream. If that happens, the mother will become sensitized against the D antigen, all right, against this positive antigen, right? And so if the mother's immune system is exposed to it and becomes sensitized against it, B lymphocytes will produce antibodies against it. And so now the mother is producing antibodies against the D antigen. What happens in subsequent pregnancies, if she has another child with the same person or really anyone else who's positive, um, what'll happen is her immune system will now recognize the D antigen present in the fetus and attack it. Okay, and so what this means is that the fetus can wind up anemic and have low oxygen carrying capacity, and that can result in death and developmental problems. All right, I think, yeah, this is the last slide. All right, so I'll leave it here. You can review some of these diseases if you wish. They're there. I'm going to turn to the screen. Do we have any questions for now? All right, well, I'm not seeing any. That's it for today's lecture. If you guys want to email me any questions you didn't ask here, I'll be happy to answer them. Uh, oh, Ashley's got questions. Okay. For Tuesday. For the exam? No, you don't need a webcam or anything. You'll be able to take it from home. It's fine. I trust you guys. At least for the first one. If for some reason, um, if for some reason I'm still with the class at the end of the semester, we'll either do it in person online or at a proctoring center. Well, to be honest, I haven't made it yet. I'll make it a little bit later today, and then I'll make a study guide for it. And hmm, it'll be mostly multiple choice. It's possible some matching or something like that could show up, but yeah, for the most part, let's say multiple choice. Is there a time limit? Let's say 2 hours. I mean, that's plenty of time, right? Bonus questions. I don't think I can do that in this format, I'm afraid. Because it's at home, I mean, I guess it's technically open book, open note. One thing you'll want to do though is make sure you don't spend too much time looking through your notes and looking through the book. That'll eat up your time. Um, so do obviously do the study guide, do study in advance, and only look up the ones you really don't know or you're really not sure about. Um, yeah, if you look up the ones you actually know, or if you don't know any of them when you go into it, you're going to be in trouble. Now that I'm not sure about. Being able to go back on questions and recheck over the test. I'm not sure. I think so. I've not actually taken a test myself through this platform before, like as a student. So, I'm not sure. I'm sorry. I think you can. Although I think it'll be a pain. I think you get one question at a time and you'd have to click back, 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 back. All right. Do we have any other questions? Is that going to do it for today? No, no, it's good. It's good to have questions. It's good to ask these. I'm sure a lot of people are wondering, and even if they weren't wondering, knowing is good. So thank you, Ashley. All right. Well, if that's it, I guess we'll go ahead and end for today. And yeah, we'll have that up later today, the study guide and the lab for today. All right. Thank you, guys.